Welcome to Affordable Tutor, a beginner's blueprint for single-cell RNA sequencing analysis. This video covers the complete workflow, taking us from FASTQ files all the way to the count matrix using 10x Genomics and cellranger. Single-cell RNA-seq differs significantly from bulk RNA-seq. Bulk RNA-seq is like blending a fruit salad and measuring the average temperature of the mixture. Single-cell RNA-seq, in contrast, measures gene expression at the individual cell level, discovering the unique characteristics hidden within the average. The 10x Droplet Engine is central to this process. By trapping a single cell and a barcoded bead inside a nanoliter oil droplet, all mRNA from that cell receives a unique 16 base pair cell barcode. This process allows thousands of cells to be sequenced and computationally sorted later. This video focuses on the final step, data processing. We'll cover everything from demultiplexing to the final count matrix generation using cellranger. This follows the initial steps of sample prep, GEM generation, cDNA synthesis, library prep, and sequencing. The core data format is the FASTQ file. Read 1 contains the 16 base pair cell barcode and the 12 base pair UMI, which identifies the molecule. Read 2 contains the actual cDNA sequence from the mRNA transcript. Index reads are used for demultiplexing by the sequencing facility. It's essential to resolve the confusion between sample indices and cell barcodes. Sample indices identify which patient or sample a read belongs to, like an apartment building address. Cell barcodes identify which cell within a sample a read belongs to, like the apartment number, and are the primary sorting mechanism for cellranger. To start the analysis, you'll need the right computational prerequisites. This includes access to a high-performance computing cluster or cloud resource with substantial RAM and storage. You also need software like SRA tools and FASTQ and a pre-built reference genome for alignment. When performing quality control using FASTQ, expect R1 reads containing cell barcodes and UMIs to fail several modules. This is normal due to their low sequence complexity and short lengths. R2 reads, the cDNA sequences should look like high-quality standard RNA-seq data, with minimal adapter content and high sequence quality. Running cellranger count involves setting several key parameters. The ID names the output folder. Transcriptome maps reads to the reference genome. Expect cells guides the cell calling algorithm. A crucial best practice is setting createBAM equals false to save significant disk space. Cellranger produces an outs directory. The essential files to keep include the web summary and the filtered feature barcode matrix for downstream analysis. Archive the raw feature matrix and ignore the large alignment BAM file, as it's rarely needed for standard analysis. Part 1 of the QC dashboard assesses recovery and depth. You should check the estimated number of cells, mean reads per cell, and median genes per cell. Good results match the expected number of cells, have over 20,000 reads per cell, and typically over 1,500 genes per cell. Part 2 of the QC dashboard evaluates signal purity and mapping. Look for fraction reads in cells and valid barcodes above 90% for minimal empty droplets and correct chemistry. Sequencing saturation over 60% indicates sufficient depth, and reads mapped to transcriptome over 70% confirms high quality. The MTX matrix format is an efficient way to store expression data. Instead of a massive spreadsheet full of zeros, the sparse matrix format only records the non-zero coordinates where a gene is expressed. The count matrix is defined by rows of barcodes, columns of features, and intersections of counts in the matrix.mtx file. When troubleshooting, look out for common symptoms. Low cell recovery can be addressed by better filtering during sample preparation. High mitochondrial content may require optimizing your isolation protocol. Use this matrix to identify root causes and actionable solutions for your experiment. In summary, success is won at the bench and preserved through rigorous quality control. The golden rules of design include targeting biological replicates, ensuring high cell viability, and achieving adequate sequencing depth. A smart data storage strategy involves skipping the large BAM files to minimize archiving needs. Finally, don't forget to subscribe to our channel and press the bell icon for latest updates.